Hi everyone. So today we are going to study regulation of respiration. So regulation of respiration refers to regulation of rate and depth of respiration according to body requirements. Regulation of respiration differs from regulation of other systemic functions for having both involuntary and voluntary control mechanisms. The involuntary or spontaneous respiration is due to rhythmic discharge of motor neurons that innervate the respiratory muscles. This rhythmic discharge of impulse from brain is from medullary and pontine respiratory centers, which in turn is regulated by chemical and neural factors. Today, we are going to deal with neural regulation. In neural regulation, we have three types of control: voluntary control, involuntary control, and reflex control. Let us see what voluntary control is. Respiration to some extent can be controlled voluntarily. This is because most of the muscles concerned with respiration are voluntary muscles. The centers for the voluntary control system are located in the cerebral motor cortex, from where the impulses are sent to the respiratory motor neuron via the corticospinal tract. Voluntary hyperventilation and voluntary apnea are possible. We know that we can hyperventilate whenever we want and we can hold our breath whenever we want. The point at which the breathing can no longer be voluntarily inhibited is called as the breaking point. Normally, it is 40 seconds. In certain conditions like bulbar poliomyelitis, and tumor of brainstem, automatic control or the involuntary control will be lost and voluntary control alone will be present. This condition is known as the Ondine's curse. Now let's move on to involuntary control. The centers for involuntary control is located in the brainstem in medulla and pons, and neurons from these area will converge on the spinal cord motor neurons. These respiratory centers are located bilaterally. They are a group of pacemaker cells, and impulse from these cells activate motor neurons in the cervical and thoracic spinal cord. The neurons that control inspiration converge on C3 to C5 motor neurons, which activate diaphragm via the phrenic nerve, and also on thoracic motor neuron that activate external intercostal muscles. The neurons that control expiration converge mainly on thoracic motor neurons that supply the internal intercostal muscles and other expiratory muscles. First, let us talk about medullary respiratory centers. We have three centers: central pattern generator, dorsal respiratory group, and ventral respiratory group. First, about central pattern generator. Medulla controls the basic rhythm of respiration. Rhythmic respiration is initiated by a small group of synaptically coupled pacemaker cells called as the pre-Bodzinger complex, which is known as the central pattern generator. It is bilaterally located in the medulla between the nucleus ambiguous and lateral reticular nucleus. The neurons of CPG in the pre-Bodzinger complex discharge rhythmically and is responsible for the normal respiratory rhythm. In addition to the CPG, the medulla also contains neurons of two types: those which discharge only during inspiration and those which discharge only during expiration. The inspiratory neurons and expiratory neurons in the medulla are concentrated in two anatomical locations: dorsal respiratory group and ventral respiratory group. These I neurons and E neurons have neuronal inhibitory interconnections and thereby exist as reciprocal innervation. Therefore, motor neurons to the expiratory muscles are inhibitory when those supplying the inspiratory muscles are active. Let us move on to dorsal respiratory group. It is so called because of the dorsal location in the medulla, located bilaterally in and around the nucleus tractus solitarius. DRG primarily contains inspiratory neurons which are controlled by pacemaker cells in the pre-Bodzinger complex. They also receive sensory impulse from lungs, chemoreceptors, and baroreceptors through vagus and glossopharyngeal nerves. Thus, it is primarily responsible for inspiration and integration of sensory information that originates from sensory system. Neurons from DRG directly project into the cell bodies of phrenic nerve motor nerves in the spinal cord. Next, is the ventral respiratory group. It is located bilaterally in the ventrolateral part of the medulla in the region of nucleus ambiguous and nucleus retroambiguous. It mainly consists of expiratory neurons. However, some inspiratory neurons are also present in the mid portion. This ventral respiratory group has three regions: rostral expiratory region, caudal expiratory region, and middle inspiratory region. Note that, okay, the middle is inspiratory region. The rostral expiratory region is also called as the Bodzinger complex, drives the expiratory activity of caudal region. Caudal expiratory region, this region in turn projects into the spinal motor neuron that innervates the expiratory muscles of respiration. Middle inspiratory region, this region turn project to the spinal motor neurons that innervate the inspiratory muscles of respiration and also to the cell bodies of neurons that innervate the muscles of pharynx and larynx. Now let us see about inspiratory RAM signal. The pattern of action potential produced by the respiratory center is called as the inspiratory RAM signal. Initially, there is a latent period, then the intensity of action potential increases and then decreases, followed by a latent period, which means initially there will be a latent period, then the action potentials increases, followed by decrease in action potential, again a latent period, again increases, decreases, latent period. This is how an inspiratory RAM signal goes on. The cycle repeated and normally duration is 4 to 5 seconds. If this inspiratory RAM signal increases, the respiratory rate will be decreased. That's all for the medullary respiratory centers. Now let us see the pontine respiratory centers. The rhythmic discharge of medullary respiratory neurons is modified by the neurons in the pons, and there are two respiratory centers, pneumotaxic center and apneustic center. Let's see about pneumotaxic center. It is located bilaterally in nucleus parabrachialis of upper pons. It has an inhibitory effect on inspiratory neurons. When it is stimulated, duration of respiratory RAM signal is reduced, thereby rate of respiration will be increased and filling volume, which means the depth, will be decreased. That is, the respiration will become shallow and deep. When this area is damaged, the respiration will become slower and tidal volume will get increased. It coordinates the respiration by switching between inspiration and expiration. It inhibits the apneustic center. Now let's see about apneustic center. It is a group of neurons located bilaterally in the lower part of the pons. They have an excitatory effect on eye neurons and prevent the switch off of inspiratory RAM signal. Stimulation of this center results in prolonged inspiratory effect interrupted by brief expiration. This is called as the apneusis. This indicates that this center strongly activates the medullary inspiratory center and thereby the duration of IRS. However, it is tonically inhibited by the pneumotaxic center. Now, let us see the role of vagus. Vagus nerve carry afferent fibers from receptors in lungs and airways. Stretching of lungs during inspiration stimulates stretch receptors in the lungs, which generate impulses in the afferent fibers in the vagus nerve from the lungs. These impulses inhibit the eye neurons in the medulla. Vagal stimulation results in inhibition of inspiration, and following vagotomy, depth of inspiration is increased. In short, switch-off mechanism coming from pneumotaxic center and vagus are the key factors which determines the rate and rhythm. Now let's see the control by higher centers. Although many centers in the cerebral cortex influence the activity of medullary respiratory center, the influence of reticular activating system and limbic system are most prominent. The reticular activating system stimulates the respiratory center to increase the respiratory drive, which is increased during the arousal from sleep. During sleep, RAS activity diminishes, which in turn decreases the respiratory drive. Arterial carbon dioxide tension slightly increases during the sleep, and this is why the respiratory drive is increased during arousal from the sleep. During emotional reaction, the limbic system also stimulates the respiratory center.
Now let us see the reflex control of respiration. The receptor of reflexes that control respiration are classified into two types. Receptors inside the respiratory system is the first one, and the next is receptor outside the respiratory system. Let us see the receptors inside the respiratory system. They are of three types: slowly adapting receptors or pulmonary stretching, rapidly adapting receptors or pulmonary irritant, and the C fiber endings. Let's see each one of them. The first one is the slowly adapting receptor or the pulmonary stretch receptors. They are located within the smooth muscles of conducting airways and lung parenchyma. They are sensory terminals of myelinated efferent fibers. They respond to airway stretch and are called pulmonary stretch receptors. They adapt slowly to the stretch imposed on them. When stimulated, they inhibit inspiration and prolongs the expiration. This is called as Herring-Brewer reflex. In Herring-Brewer reflex, we have Herring-Brewer inflation reflex and Herring-Brewer deflation reflex. First, let us see Herring-Brewer inflation reflex. What happens? When there is increase in tidal volume more than 1 liter, the stretch receptors of the lungs are stimulated. As a result, there will be increased discharge of efferent impulse to the inspiratory center through the vagus. And there will be inhibition of inspiratory center because the tidal volume has already reached 1 liter. Again, we don't need inspiration because the lung is already very much inflated. So there will be inhibition of inspiratory muscles and increase in expiration time occurs. The reflex operates only when the tidal volume exceeds 1 to 1.5 liter as in excess. Thus, it prevents the overinflation of the lungs. The stimulus in this case is the distension of the lungs, and the receptors are the slowly adapting stretch receptors in the lung parenchyma and in the smooth muscles of airways, that is in the trachea, bronchi and bronchioles. The efferent limb is the vagus and the efferent limb are the nerves supplying the respiratory muscles, especially the expiratory muscles. The center is the inspiratory center in medulla. As a result, the effect is inhibition of inspiratory center and switching off of the IRS, leading to expiration. Thus, lung volume is brought back to normal. Let's move on. Next is the Herring-Brewer deflation reflex. In this case, there occurs the severe deflation of the lungs. So, no stimulus of the stretch receptors of the lung. No discharge of efferent impulse to the inspiratory center through the vagus. The IRS will be prolonged in this case. As a result, increased inspiration occurs and increase in filling volume will bring back the volume to normal. Here, the stimulation is over deflation of the lungs. The receptors are slowly adapting stretch receptors of the lung parenchyma and in the smooth muscles of airways, that is trachea, bronchi and bronchioles, same as in the case of inflation. Here the efferent and efferent are vagus. Center is the inspiratory center in medulla. Effect is no inhibition of inspiratory center and prolongation of IRS, leading to increase in inspiration. Thus the lung volume is brought back to normal. Now, let us see the significance of Herring-Brewer reflex. This is a protective effect that protects lung from damage due to overinflation while maintaining normal alveolar ventilation. It decides the filling volume of the lungs, especially in conditions of hyperventilation, as in the case of exercise. It is more useful in infant because it is used to maintain tidal volume during normal breathing in case of infants. Now let's move on to rapidly adapting receptors or pulmonary irritant receptors. Here it is the sensory terminals of myelinated efferent fibers found in larger conducting airways between the airway epithelial cells. Here the stimulus is hyperinflation of the lungs. Exogenous and endogenous substances like histamine, prostaglandin, smoke, dust, irritant gases like SO2, etc. They are stimulated by irritant and thus the name pulmonary irritant receptors. Responses are increase in firing rate, hyperventilation, bronchoconstriction, coughing, sneezing, etc. What is the significance of rapidly adapting or irritant receptors? They play an important role in detecting pathological processes that involves irritation, congestion and inflammation of airway. They help to remove noxious agents from tracheobronchial tree, and bronchospasm is beneficial for prevent further entry of noxious agents. Here in this we have cough reflex and also sneezing reflex. In cough reflex, it is the stimulation of irritant receptors of larynx, trachea and bigger bronchi producing the cough. Coughing begins with a deep inspiration followed by a forced expiration against a closed glottis. Glottis suddenly opens and air is expelled at very high velocity. Excess coughing will lead to alveolar rupture, visual blackout, fainting attack, herniation, etc. Sneezing reflex, it is stimulation of irritant receptors of nasal cavity and upper airway produces the sneezing. The last one is the C fiber endings. They are terminals of unmyelinated nerves. There are two types, pulmonary C fibers or juxtapulmonary capillary receptors or the J receptors located in alveoli which are in close proximity to pulmonary capillaries. The other one is the bronchial C fibers located in the airways. Both type of C fiber receptors are stimulated by hyperventilation, injury, acute pulmonary vascular congestion. They also respond to intravenous and intracardiac administration of chemicals like capsaicin. The reflex response produced is apnea followed by rapid breathing, bradycardia and hypotension called as the pulmonary chemoreflex. A similar reflex produced by receptors in heart is called as coronary chemoreflex or Basel-Jarisch reflex. Its significance is that physiological role is uncertain. Probably it can occur in pathological state such as pulmonary congestion or embolism in which it is produced by endogenously released substances. And the last one is receptors outside the respiratory system. There is those which stimulate the respiration and those which inhibit respiration. Those which stimulate respiration are proprioceptors, nociceptors, thermoreceptors and emotional stimuli from hypothalamus and limbic system. Those which inhibit respiration are baroreceptors and visceroreceptors. That's it for the neural regulation. I know there are so many topics and there are subtopics among them. So don't miss out any headings. Hope you guys understood this. Thank you.